Assalamualaikum and hi, my name is Umi Ailina binti Kamaru Amri. My metric number is 2020-971-839 and I am from M380-2225A. Today, I'm going to share with you about the application of seed priming in crop production under the code AGA 602, which is seed technology. So for the introduction, seed priming is the process of regulating the germination by managing the temperature of the seed and also the seed moisture content, and also the seed will undergo the biochemical treatment process. Seed priming, also known as a pre-sowing seed treatment that allows to control the hydration of the seeds to absorb the water and also go through the first stage of germination, but does not allow the radicle appear through the seed coat. So when we are going to apply the seed priming in the crop production, so the situation is that when we are being the owner of the greenhouse, we are planted the bedding crop. Then suddenly there's a client who order 100 flats of the begonia seedlings. We will be panicked because the begonia seedlings are slowing in germination. So by applying the seed priming in the crop production, we can fasten the germination of the begonia seedlings, and the customer also will be happy and repeat the order with us. So for the mechanism of the seed priming in crop production, seed priming allows the regulation of the water content in the seed. Either it will be soaked in the water of the solute or it will expose the seed into the water vapor. So second, the priming process regulates the seed's temperature and also the moisture content, bringing the seeds closer to the point of the germination. Third, for treatment that using water, the seeds absorbs the water for a predetermined time interval. After the time interval, the process is halted right before the first root. It will be called the radicle to emerge from the seed. A high amount of the water is needed for radical emergence, so the priming process is key to prevent the full germination from occurring. So as you can see, the green line refers to the prime seed and the blue color refers to unprimed seed. It's divided into three phases, which is phase 1, seed imbibition, phase 2, metabolism activation, and phase 3, growth cell elongation, which is the radical protrusion. When the seed undergo the priming treatment, the seed able to become dehydration and manage to undergo the storage process compared to unprimed seed. When the seed not undergo priming treatment, the seed will not be able to put up in a storage. The cell elongation also increase and make the seed has increased rate in terms of the germination. It also depends on how much water content inside and following the time. There's another example of comparison between prime seed and unprimed seed. When the seed undergo the imbibition in matrix solution, the prime seed is able to undergo faster emergence and manage to germinate more early compared to non-prime seed. If we look to the diagram at B, we can see the differences between the prime seed and non-prime seed. The prime seed grew in uniformly compared to the non-prime seed. This is the corn seed that we use on the tray. Then after that, the seeds can be dry and sown when the seeds are already fully ready. So there are 4 types of the seed priming in crop production. The first is the hydropriming, the second is osmotic priming, the third is solid matrix priming, and the last but not least is drum priming. So the first thing is the hydropriming. Hydropriming is the process when we soak the seeds in the water, and this method is more economical and also easiest to do. For the example of the hydropriming, We took sorghum plant as a sample. The parameter for this experiment are the yield rate and also the fungal infection on the seed. Basically, the treatment is all about soak the seeds in the water. We put the sorghum seed with no treatment and undergo hydropriming treatment. The rate of sorghum with hydropriming treatment produce more yield even though with the same rate of fungal infection. This way are more better compared to not putting any treatment to the sorghum seed. The second is the osmotic priming. Osmotic priming is the process when we soak the seeds in the solution such as chemical, and mostly the chemical that has been used is KCl, KNO3, and also NaCl. By applying the osmotic priming, we can see the differences on the surface of the seed coat. It will increase the air-filled spaces between the cotyledons and also the embryonic axis. Besides, it will expose smoother and flatter seed coat surface and also the existent appearance of microcracks inside the pit. It will also increase the vacuolization level that bring changes in expression pattern of aquaporin genes. For the post-priming germination, it will also contribute to high kinetics of water uptake and improve the water status of the seeds. This may also add plant hormones and also beneficial microorganisms. Third is the solid matrix priming. Solid matrix priming is the process when the seed will incubate solids such as vermiculite with small amount of water to allow to absorb in the seed. During solid matrix priming, seeds are mixed and incubated with wet solid water carrier for a certain period. Afterwards, seeds are separated from matrix, rinsed and back dry. The use of solid medium allows seed to hydrate slowly and simulates natural imbibition process occurring in the soil. As we can see, for solid matrix priming, the green line refers to the prime seed and blue line refers to non-prime seed. Following the movement of time, the prime seed takes short time in order to have high percentage of germination. Prime seed has decreased range of germination times and shift germination forward in time. And last but not least is the drum priming process. Drum priming process is the process when the seed are hydrated by placing them into the rotating drum into which a control level of water vapor will be released. So these are all the important components in order to undergo the drum priming treatment. For the example, motor, timer, drum, solenoid, and tank. Drum priming treatment is different compared to other treatment because this is only the treatment that use the technology inside. The overall consequence of seed priming consists in an increased seed vigor defined as the whole set of properties conditioning seeds' lots performance in a wide range of the environment. Priming strategies may afford several economic and agronomic advantages to the cultivated plants. So there are advantages of the seed priming in crop production. The first is the seed priming fastens the emergence of the germination. They also enable the seed to germinate and emerge even under adverse agroclimatic conditions such as cool and wet or extreme heat. Seed priming also helps to optimize harvesting efficiently, which increase the yield potential, and also helps to improve vigor for faith and healthy plant development. But every advantages they also has a few disadvantages. The seed also are difficult to store in some cases as they need cool storage temperatures. They also increase the dependence of the chemical to the agricultural land, and last but not least is dependent upon crop or variety specific close optimization of the studies. So that's all for the seed priming. I hope this will help all the people outside there. Thank you.